All right, we are ready to go.
Okay. Well, I'll introduce myself. You all know who Danae is, our Executive Director of HCU Network America, who coordinated this webinar. But I'm pleased to be able to share this information with you. As I will explain as I go through this, I played a pretty significant role in pulling this information together, so I'm pleased to present it to you. If you go to the next slide, Danae. And so, next slide, please. Okay. So the first slide said, I am the president of the board of HCU Network America and one of the co-founders along with Danae. And about 18 months ago, we undertook this project with HCU Network Australia. Excuse me, I'm going to move my husband's iPad away from me because he has a barking dog. Hold on one second. Okay, his iPad is now nowhere near my speaker. Sorry about that. He left the room, but his iPad didn't. Anyway, we started this project across our two organizations because we wanted to understand all of the research that's going on. So I'll tell you how I went about it. But I always believe that it's important for anyone who has different roles in a given area to make sure that they're totally transparent about that. So I also want you to know that I am on the board of Orphan Technologies, which is a company that does have a product in development for HCU. Now, the way I got on that board was I had funded some of the work originally done on that project through a foundation I set up, the Hempling Foundation for Homocystinuria Research, which I started in honor of my sisters who passed away from homocystinuria in the 1970s. On the other hand, I made clear as I joined the board that I needed to continue my impartiality. My goal is to get as many products on the market as we can to help patients with homocystinuria. So I continue to fund research across several of the areas we'll talk about today, as well as I donate any board fees from Orphan Technologies to HCU Network America, so I don't have any financial gain from that activity. My goal is to help the company get their product to market to help HCU patients. So if you go on to the next slide, Danae. So Tara Morrison and I started this project with the goal of developing what's called a research map, and this is a term that's used by other organizations who want to understand all of the research going on in therapy. Our focus was homocystinuria caused by CBS Deficiency. We do intend over time to look beyond just CBS Deficiency. And so we also wanted to develop a research strategy and get input from experts in the field. What do they think the key priorities should be for this area, and use that to help guide funding of research. And I'll talk about a global grants program that we have started in collaboration with HCU Network Australia. Next slide, please. I hired a consultant to do this work, so we had someone that had the expertise in genetic diseases, rare diseases, to understand and to be able to search for all of the information that's in the public domain, that's been in different journals, that's abstracts presented at meetings, etc., as well as information that was provided to her by academic and industry researchers. We supplemented that with interviews with key global experts, both clinicians who treat patients with homocystinuria, as well as PhD researchers who may do more lab work in trying to develop new therapies. And there are some we interviewed who would fit both of those categories who were especially helpful. Next slide, please. You went backwards, Danae. If you could go forward now 2 slides. Okay. So I mentioned that we had the consultant interview several experts, and all of the academic clinical experts are listed on the left part of the slide. I won't read you the names, but you probably will recognize some of those names from presenters at our patient meeting in April, or names that you've seen appear on website, Facebook, publications, etc. We also interviewed individuals in the industry who had specific programs. Our interviews with them focused on their own program. We didn't ask them to evaluate their competitors' programs, and I know from having a background in the pharmaceutical industry that you might find them most excited about their own programs, which is really just a function of they really believe in what they're doing. Next slide, please. So you've seen this pathway, and I'm using this to set up an explanation of how the different products I'm going to talk about fit into the biochemical pathway. Clearly we know that homocystinuria is caused by an excess of homocysteine in the blood. And homocysteine is made from methionine that we all take in through protein in our diet. The CBS enzyme is involved in metabolizing homocysteine to cysteine, which then goes on to other compounds. So there are various pathways by which you could influence all of these compounds, and you're going to hear how the different products in research fit in. So if you go on to the next slide. So this slide lists different areas, and somehow the numbers got a little mixed up. Imagine that says 1 through 5. These therapeutic approaches are all being studied to treat CBS deficient HCU. And so I will go through each of these in greater detail, but this is just a high-level summary of the different approaches. So how can you lower plasma homocysteine? Well, one approach is you could replace the enzyme that's defective. And there's a product in development that's called enzyme replacement therapy that replaces the CBS enzyme. So I'll go into more detail on that. Another approach is you could actually deliver the DNA to enable cells to produce the CBS enzyme, or you could deliver bioengineered red blood cells that contain the DNA, and I'll tell you a little bit more about that. Another approach is you could use alternative enzymes that can degrade either methionine, so you don't convert that methionine to HCY, or you could directly degrade homocysteine. Another approach is to activate the protein, and enzymes are proteins. And if you have a faulty enzyme, there may be compounds that could restore that enzyme function or activate that enzyme activity. And lastly, you can modify the metabolic pathway through either nutrition or administering different compounds that can address the different metabolic imbalances. So if you go on to the next slide, I now show you, going back to this biochemical pathway, how the different categories of products I just talked about fit into the pathway. So up at the top left, you could have an alternative enzyme that breaks down the methionine, so you have less methionine to convert to homocysteine. You could modify the metabolic pathway, which could directly affect methionine, or it could affect the conversion of methionine to homocysteine or homocysteine back to methionine. You could also administer an alternative enzyme that directly breaks down homocysteine, an enzyme different than CBS enzyme. You could also, at the bottom left, modify the metabolic pathway through a few different compounds we'll talk about. And then the bottom right, we're showing 3 different approaches to directly affect the CBS enzyme: replacing it, delivering the DNA to make the enzyme, or cellular therapy that has the DNA, or activating the protein or enzyme. So I'll come back to this at the end once more, but I thought it would be a useful schematic to help you see how these products fit in. You go to the next slide, Danae. So I'm going to use the same setup on this slide to go through each of those 5 categories. And I will talk about a little bit more about the approach and what the status is, who the sponsors are. And then I will give you the perceived strengths and benefits, weaknesses, or limitations. And that's all based upon input from experts. You really won't know until these products get through a development cycle and until they're approved by regulatory agencies, what to assume about efficacy and safety and dosage and administration, and certainly you don't know price until these products are marketed. But these are all impressions that the experts had, and I'll let you know if some were outlier opinions, or I'd say the majority of them were pretty consistent across the experts. So let's start with the CBS enzyme, and the name of it is OT58. Companies very often will have an acronym that is consistent with their name, in this case Orphan Technologies, and the project number is 58 for this product. And PEG in front of CBS is because it's a pegylated version of CBS, which is another structure that's attached to the enzyme that helps give it all the right characteristics to be stable, to have the right blood levels, etc. So this is a project that originally came out of Jan Krause's lab at University of Colorado, and that's when I first became involved through the foundation I set up in honor of my sisters. I provided a grant which funded a mouse study which generated proof of concept. Some of you may have seen the publication or reference to it, that administering this enzyme, and the enzyme was made in E. coli bacteria by using recombinant DNA, and that's a technique that's often used to create biologic products. And so Jan had developed this, and after he had positive data, it was licensed in by Orphan Technologies, and Jan stays involved in helping to do a lot of the work to help get this product to market. And so this is, as I said, a manufactured version of the human CBS enzyme that takes the DNA for human CBS, and the E. coli then transcribes that DNA or expresses that DNA to make the enzyme. This is expected to be given by subcutaneous injection, and I believe that's once or twice a week. Both approaches were being tested. And the phase 1, which is the first stage of human testing, is going to begin this quarter. And I haven't heard yet if the first patient has been enrolled, but I was told that would likely happen in the month of October, either at Children's Hospital of Pennsylvania or Boston Children's Hospital. And there are a few other sites involved. You can see the details of those sites in the research section of our website. And certainly if any of you live in those areas and you yourself or a child could potentially qualify for the clinical trial, you can look at our website or reach out to Danae, and she can steer you to the right place to get further information. And we'll do that for any clinical trial, for any product that's being developed for HCU, because it's in our community's best interest to make sure that we get good participation and support in all of these trials. So what do the experts think the strengths and benefits of this approach would be? Well, first, it's designed to lower homocysteine, the most important benefit, and should therefore achieve the same effect as dietary restrictions if you are able to get anywhere near the efficacy that you saw in the mouse model. And so over time, that could lead to relaxation of dietary restrictions. I don't think doctors would tell you to do that immediately until you saw how the enzyme was working, but that could certainly be something they may try over time. In mice, the homocysteine was lowered to near normal levels. And another benefit of this approach, especially vis-a-vis some of the others we'll talk about later, it would be expected to work for all CBS patients, regardless of the underlying genetic defect. So there's close to 200 different defects that have been discovered to cause CBS deficient HCU, and it wouldn't matter what defect it was, this should work. What are the perceived or potential weaknesses and limitations? There are other enzyme replacement therapies on the market, for example, for Pompe disease and Fabry disease. And if the cost of this product is similar to those, payers may question the cost-benefit ratio. Those products, though, are generally reimbursed by insurance companies in the U.S. as well as paid for by national governments internationally. Second, as I said, it's a subcutaneous injection, so you wouldn't need to go to the physician's office to administer that, but a patient would have to be willing to administer it to themselves, or a family member would have to help, especially a child, with that administration. But as many of you may know, that's a lot easier than intramuscular injections or intravenous infusions. The next potential concern, enzyme replacement therapy is not always effective in humans. Sometimes that's because you get an immune reaction where your immune system develops antibodies against the enzyme, and rarely these other products have been known to have severe anaphylactic reactions. So that's why you need to go through extensive testing before you can get on the market to understand whether that happens and how big a risk it is. So let's move on. That was it on that slide. Let's move on to the next slide, and we'll take a lot of questions at the end, but certainly if I'm not being clear, well, I guess, Danae, we don't have the lines open, so we'll wait till the end for questions. So the second approach is to use either gene therapy or cellular therapy, and I'll go through each of those individually and try to explain the difference. For gene therapy, the way that works is you would take a normal copy of the CBS gene and you would insert that into a vector. And a vector is just a delivery system. It's feasible to just take DNA and inject it, but it doesn't work real well. You have to actually give an electric shock to someone if you'd want to administer it that way, which I know of trials that have been done using that approach for very severe disease or very serious diseases for which there's no other approach, but I don't think anyone wants to do that. So a vector, and in this case they use an AAV or adeno-associated vector, which is related to the cold virus, but it has no biologic activity. So that's really a carrier vehicle that enables you to inject the DNA into your cells to enable your own cells to express the gene or express that DNA and to produce the normal enzyme. So I like to say it turns your own cells into a manufacturing plant that you have the DNA you need to make the CBS enzyme and you just keep making it. So it's a novel concept. It's being tried now across many rare diseases. When I attended the expert meeting for metabolic diseases last year in San Diego, I think I heard about 9 or 10 different metabolic diseases that gene therapy was being tried for, including PKU. And so there is one program right now that's still in progress, and that's being done by Warren Kruger at Fox Chase Cancer Center, as well as Ron Crystal at Weill Cornell in New York. His lab is making the product, and Warren is testing it in his mouse model. And I've also provided funding for this proof of concept study through the foundation that I have in my sister's memory. And Warren expects this study to be completed by the end of this year, and so we look forward to data on that approach. So what do the experts think the potential strengths and benefits could be? First, this could be, quote, curative if the gene enables replacement of the enzyme and it lasts lifelong. And it's unknown at this point how long it will last. I'll come back to that. Certainly, if you had enough enzyme, you could lower or totally eliminate dietary restrictions. And there have been recent approval of gene therapy for other genetic diseases. So this is starting now to be an area where the more testing that's done, the more comfort level there is. And so I think over the next 5 or 6 years, you're probably going to see upwards of 20 products in development or coming onto the market for severe genetic diseases. And so retinal dystrophy is an example of one that a company in Philadelphia called Spark Therapeutics recently came out with a product for, and it's restoring vision in patients who had a genetic form of blindness. So what are the potential weaknesses and limitations? Well, those of you who have knowledge or are well-read in the medical area know that gene therapy has been talked about for 20, 30 years, but it's still very much in early stages. And so the long-term risks and efficacy are not fully established. Some of the patients for other diseases have been followed now for upwards of 5 years without untoward effects, but you certainly want to follow even longer. not just to see if there are safety risks, but also to make sure that efficacy continues. It's unknown if you'll maintain high levels in your cells of the DNA. Cells turn over. Do you lose enough cells that you're not making enough enzyme? And so it could require readministration over time if that were to happen. It's possible this wouldn't work for all CBS patients, but I think that was an outlier view. I think if the gene gets replaced, there's a good chance it would work for everyone. Based upon other gene therapies, payers, again, will likely consider the cost to be high, and they have to be willing to accept high upfront payments but avoid the annual fee. And I know the first product that was introduced for retinal blindness, the cost was somewhere in the $500,000 range, but they didn't expect payers to pay for that all at once. It was an upfront payment and an annuity over time. So it could end up looking more similar to other products that have a high annual cost, but that will play out over time, and we'll see where that goes. There's a technical risk that if you have to re-administer the product over time, you may have to switch the type of AAV vector. There are different numbers, for example, AAV8, AAV9, AAV2. And if your immune system develops antibodies to the original vector, you wouldn't want to re-administer that. So that's something that would be tested for, and if the manufacturer saw that antibodies were developing, they would have to get a different form of the vector ready for re-administration over time. Next slide, please. So what is cellular therapy? It's something that's being used now for severe rare diseases as well as for severe forms of cancer. And in the case of HCU, there's a company that has developed a genetically engineered form of red blood cells. And they insert DNA into those red blood cells for the CBS enzyme. And the theory then is that the red blood cells will make the CBS enzyme, and when homocystinuria is taken up by the cells, the enzyme will metabolize the homocystinuria and release the byproduct. So it's almost, again, like having a little manufacturing machine or plant, within your own cells that do that work. And so there's a company in the Boston area named Rubius Therapeutics, and if you look on their website, you will see the acronym RTX CBS. And they state that the status of their program is lead optimization, which means they're testing various variations of the product to see which one works best before they decide to go further forward into animal testing and animal safety studies, and then into humans. So that's a little bit further behind, certainly, than the first program we talked about. So potential strengths and benefits. Now, for this program, because it's a fairly new program, we just became aware of it this summer. It was not included in the interviews done about a year and a half ago with the experts. So you're getting the personal views of myself, but I will say I do believe I have a pretty good understanding with a pharmacy background and 30 years experience in the pharmaceutical industry. But I'm certainly not an expert. Like over time, we will get our true experts to evaluate this. So you can take this with a grain of salt. So potential strengths. If it's effective, it could lower homocysteine levels and, again, reduce the need for dietary restriction. That's pretty much true for anything that works in this field. It could potentially avoid autoimmune reactions that may occur with enzyme replacement therapy or AAV-delivered gene therapy, but that would have to be proven. It could potentially be administered less frequently than enzyme replacement therapy, which, as I said, would be once or twice a week. This product, because of the amount of time that a red blood cell stays in your system before it turns over, could potentially be every 90 days. You'd have to get a new short infusion of red blood cells. The other potential benefit is that this program for HCU will benefit from the company's experience with a similar program for PKU. And they are expecting to begin human trials for PKU next year. And so while we're a little bit further behind, sometimes that enables you to learn from what happens with the first program and have a faster, smoother approach. So potential weaknesses and limitations, again, my perceptions, and certainly the efficacy is not yet shown. Theoretically, it makes a lot of sense, but you need to see how much uptake do you have of homocysteine by the red blood cells, and do they actually express the DNA and produce the enzyme and metabolize the homocysteine? So a lot of unknowns until you get further data. Safety is unknown. Also, you'll probably need to cross-match the red blood cell type, and so there could be a small percentage of patients that may not qualify based upon the red blood cell type, but that's expected to be small. Again, would require a short IV infusion, and while 90 days is theoretical, that has to be shown through subsequent trials. And other cellular therapies are considered to have a high cost, so that will certainly be determined over time. There are certain therapies being used for cancer where you actually take cells out of the individual and then bioengineer those cells and put them back in. That's a much costlier process, and this would not be as costly to manufacture, you know, the bioengineered red blood cells. You wouldn't expect it to be, but until we get further along, we really don't have any idea on that. So I know that was a pretty in-depth technical explanation, but I'm happy to answer questions on it later. So let's go on to the next slide. So alternative enzymes. There are two of these enzymes I'm going to talk about, both of which have company sponsors. The first is called erimethionase, and that is a project that was in place by a European company called Eritech, which is based in Lyon, France. And they are doing work on methionase, which is a bacterial enzyme that they also encapsulate into red blood cells and give per IV infusion. And it degrades the methionine and lowers, believed to lower both methionine and homocysteine. And that has been shown, studies conducted in mice, but the company currently has the program on hold due to other priorities they're working on with their enzyme program. But they may come back to it at some point in time, and they promise to keep us posted. Potential strengths and benefits. Again, it's expected to lower methionine and subsequently homocysteine, so that should mimic the effect of having a protein-restricted diet. So you could potentially relax dietary restrictions. And if it works, it should work for all CBS patients. Potential weaknesses, you'd have to give an IV injection, and they believed it would have to be monthly. It is a bacterial enzyme, so it could trigger an immune response in humans, which could cause allergic and potentially severe reactions. And it doesn't restore the function of CBS, so you don't correct the downstream metabolites, and it's unknown what the implications of that would be, but it is hypothesized that some of those downstream metabolites are important in causing symptoms of homocystinuria. And again, the potential cost of this is unknown. So let's go to the next slide, and there's another alternative enzyme that's called homocystinuria, and the acronym for this is from a company named Aglia, which is in Texas, is AEB 4104. And they actually just presented an abstract at a human genetics meeting over the last week, and so you can do a Google search or go to their website and see that abstract. So it's a recombinant human enzyme, which means they make it, through DNA. They actually take a different enzyme and they introduce some mutations into that enzyme, and it's given via IV infusion. And it's expected to directly degrade homocysteine, therefore lowering homocysteine and potentially methionine. And so in the proof of concept study, which is what was in the abstract presented over the past week, it did significantly reduce homocysteine, it stopped progression of the disease, and it improved survival of the mice that were studied. So the potential strengths and benefits, and these interviews were done before the recent data were available, but I'm not sure it would change it dramatically. But it would be expected to lower homocysteine in humans and mimic the effect of dietary restriction, which therefore should also lower methionine levels, should work for all CBS patients, therefore reducing dietary restrictions. And one of the experts thinks it could be very useful if you need to very quickly lower homocysteine levels, such as you have a patient, a homocystinuria patient who needs surgery and has very high levels, which could cause a clotting risk. So this could be a useful acute approach if studied that way. Potential or perceived weaknesses, the IV injection would require you to go to the hospital or physician's office at least monthly. It could trigger an immune response with severe reactions possible. Like the prior slide, it doesn't restore the function of the enzyme, so it doesn't help correct the downstream deficiencies. And again, its potential cost is unknown. So let's go on to the fourth category. And I'm not going to spend a lot of time on the next couple of slides. These projects don't have a lot of activity going on, and these programs are believed to not be applicable to all CBS patients, so would be less useful for the community. But theoretically, it's possible to help restore that enzyme activity. Compound by Moclamol. Warren Kruger does have a NORD grant to study that in combination with another product, bortezomib. So we'll see if we get data out of that, but I know some of the earlier studies did show limited efficacy of the monotherapy. So let's go to the next slide. We also have products called proteasome inhibitor, which help present the breakdown of whatever enzyme you do have. And products like this are in development or on the market for cancer, but it would only work for defined mutations where you still have enough enzyme that preventing it from being degraded could yield a benefit. And bortezomib is the compound I mentioned that Warren Kruger has a NORD grant to study. It is believed to work in mice with CBS deficiency based upon some earlier work. It is available for other indications, but it does require frequent IV infusions, and it's not perceived to be safe for chronic use. So the real hope with something like this is there might be another similar type of product that is safer that could be developed, but I personally think it's more of a long shot. Let's go on to the next slide. And then there's a third category. There are compounds that can stabilize the regulatory domain, which could help with refolding of the enzyme or activating a mutant enzyme. And I won't try to explain the technical aspects of this, but there is a program going on that's in research stage that's going on by Thomas Machin at University of Colorado. And there's an American Heart Association grant that he has. So we'll see if he comes up with any data that would lead to greater activity in this area. So let's go on to the fifth and last category. And this category includes some different approaches to alter the metabolic pathway. And the first would be sulfur amino acids dietary modulation. I won't go into that because it doesn't have a sponsor. And Victor Kosick, who did this initial work, thinks it would be very hard to develop this approach for regulatory approval. A second approach is taurine supplements, and some of you may have been aware of or potentially involved in a short-term clinical study done with taurine that was sponsored by University of Colorado. And taurine, by the way, is contained in Red Bull drink. So if any of you are really excited about this, but I wouldn't encourage you to try it on your own. Really hasn't been that well studied. But It is orally available, it is a low cost, and they did have data in the pilot study that showed that there was a positive effect in helping CBS patients who had abnormal endothelial function, and that relates to clotting in the blood vessels. But that would need to be replicated in a much larger study, which is being considered, but it's unknown if that will have enough support to go forward. It's also unknown if this positive benefit would occur for all CBS patients or if it's limited to those with that abnormal function, which you would then need to test to know who it could be useful for. So let's go on to the next slide. And this last project, Formate, this may be the first time many of you are hearing about this. I wasn't aware of it until a few months ago. This is a project going on at University of Colorado by Ken McLean. And Ken has been receiving funding from the Will Homo Foundation, one of the HCU families in the U.S. Has a fund that they've been supporting Ken's work in the past. And we became aware of it because I'll mention in a moment, we initiated a global grants process, and Ken submitted this proposal, and it received favorable reviews from the Scientific Advisory Board and the expert reviewers. And we actually have offered a grant. It says here potential grant because we're waiting for approval by the university and Ken. And then once we have that, we will formally announce this via press release, so stay tuned. And so what this would entail is administration of Formate, which is a substance that our bodies makes on its own, but you'd administer here a higher level than would normally be made by an individual. And combining that with betaine could dramatically lower homocysteine based upon a mouse model. And so a preclinical, which means animal study, to generate proof of concept in a CBS deficiency model, we don't yet have that, is expected to begin this quarter with potential funding not only from HCU Network America, also HCU Network Australia. We are supporting this jointly, but also, as I said, funding from the Will Homo Foundation. This program, like the other previous one I mentioned, has not yet been evaluated by our expert panel. So these are my own personal views, but certainly, like other compounds, if it works, it could reduce dietary restrictions. When combined with betaine, we have no evidence it works well enough on its own. That could certainly be tested over time. And since Formate is made naturally by the body, there could be less chance of side effects depending upon the dose that's needed. Potential weakness, we don't yet know the efficacy and safety in humans. It may not work in all patients. Dosage and frequency of dosing is unknown. It would still require betaine administration, and the cost of this is also unknown. So stay tuned on this one. You'll be seeing hopefully a press release from us announcing the grant, and then hopefully within a year or so we should have data out of this proof of concept study. So let's move on to the next slide, please. So now I'm going to circle back to the biochemical pathway. And I showed you before the category of approaches that addressed the different steps of the pathway. I'm showing you here the specific names of the projects, the methionase enzyme, the sulfur amino acids, both impacting the methionine step. Formate impacting at the methionine to homocysteine, homocysteine back to methionine along with betaine. Homocystinase helping to directly metabolize homocysteine. Taurine working to restore some of the biochemical deficiencies, the lower end of the pathway. And then the approaches to directly replace or activate the CBS enzyme, enzyme replacement therapy, gene therapy, cellular therapy, chaperone compounds, proteasome inhibitors, and products that could stabilize the regulatory domain. So I know this is a lot of technical information, but hopefully the concepts are clear enough to give you a sense that there's a lot of work going on, all different approaches affecting different parts of the pathway, are being assessed, and I think we should all be hopeful. There has not been a point in time when this many projects were in the works for homocystinuria. Some of this is enabled by new technologies like gene therapy and cellular therapy that are being developed. Some of it is enabled by the availability of grants, which we're helping to increase, and some of it hopefully is being supported by the HCU community, not just in the U.S. but globally, that's encouraging more research around the world. So let's go on to the next slide. So we asked at the end of the interviews for the experts to give us their guidance. If we really want to make a difference for the HCU community, what do they think the key priorities should be, both for future research but also patient advocacy? And we broke down their responses into 3 categories. The first, optimize current therapy. Make sure we take what we already have and know what long-term outcomes can be expected. Let's improve the quality and the taste of the different foods. Let's better define the approach with supplements, who needs them, at what dose, at what frequency, and let's better understand the impact on quality of life. And then improving detection of CBS. Certainly if we can improve newborn screening, we can help to lower homocysteine levels before any symptoms occur. But once symptoms occur, we've heard many examples, some of you may have your own, of how long it can take to get an accurate diagnosis. Let's make sure we speed up that process. And let's make sure overall that there's an improved understanding of how common this disease is and an overall awareness of CBS deficiency so that the community can get better support, not just from healthcare providers, but in general. And then lastly, the area that we were most focused on with the project, what new treatment modalities did they see as highest priorities? And again, this was about 1.5 years ago. It'd be interesting when we next do this project a few years out to see how this changes, but they were most excited about safe oral therapies that could help ease dietary restrictions, like chaperones, although, as I said, that we haven't had promising data like we've had in other categories. They were excited about enzyme replacement therapy with either the CBS or alternative enzymes so that you can directly lower methionine or homocysteine. And they were excited about gene therapies, including gene transfer, but potentially now we have cellular therapy, with gene therapy being perceived as a potential one-time curative approach. Next slide, please. So I've mentioned that we've initiated a global grants program. So after we completed this project, we had a research report developed, and we had a set of slides developed which are similar to what you're seeing today. We then convened a scientific advisory board and held a meeting at the Rio global meeting of metabolic experts last September. So our scientific advisory board came together, reviewed the output of this project, reviewed a similar set of slides, debated what they thought of the different approaches, but came to agreement on issuing a call for expressions of interest. And we identified 2 areas that these potential grants we would provide could cover. First, an improved approach to newborn screening, and second, a novel approach to treatment. They did not want to try to pick a winner. They felt that all of these different approaches could yield a safe, effective therapy, and we won't know until they go through the development and regulatory cycle which ones will make it. So let's see what proposals come in and pick what we think is the most innovative approach. So we then issued that call. We reviewed the proposals that came in and with our scientific advisory board, and then we requested full proposals from 2 applicants in March of this year. Those proposals were submitted along with the budget. They were reviewed by our scientific advisory board as well as external reviewers who had a high level of scientific expertise in each of the 2 areas. As a result of this process, we did award 1 grant or offer 1 grant to Ken McLean, as I mentioned, pending final sign-off. and that would be for a total of $40,000 US, and that's split between HCU Network America and HCU Network Australia. And from HCU Network America, it's primarily being funded by the Hempling Foundation for Homocystinuria Research. Ken also will access funding from the Will Hummel Foundation, so we're appreciative of that support as well. And what his study will do, as I mentioned, it will look at formate combined with betaine in a mouse model to generate proof of concept. The second category, the improved approach to newborn screening, we had one really solid, innovative proposal, but there were several technical issues identified by the reviewers, and so we've asked that researcher to resubmit that proposal addressing those technical issues in the next round, which we are planning for the first half of 2019. Next slide, please. So where are we going from here? We hope over time to increase fundraising through our patient groups and community so that we can support even more of this important research. We will continue to liaise with industry and academia to support increased research. Whenever we find out that there's a company involved in the field, we reach out to them. We make sure that both HCU Network America, HCU Network Australia, and other groups that we've started to partner with internationally are aware of what is going on. We have discussions with the industry or academia experts. We offer support to help with enrollment in clinical trials, etc. So we really want to support each and every program that's out there with the end goal of getting more helpful therapies for patients affected by homocystinuria. We also hope over time to go beyond CBS deficiency. This was a lot of work, cost a fair amount of money to hire the consultant, develop the report, convene the SAB, and implement the grants program. So we wanted to focus early on to generate some momentum. Next slide, please. So I know I've covered a lot of information, and I will take questions, but first, I want to give some big thank yous. This was a lot of work, a lot of effort, and I had a lot of help from a lot of good experts. First, we had a very talented consultant who's an MD, PhD with a lot of experience in rare disease research, Marion Howard. Tara co-sponsored this project with me and helped every step along the way. We had a scientific advisor for the project, Victor Kosick in Prague, who also chairs our scientific advisory board. And then I list all of the experts that were interviewed, and then I list the members of the scientific advisory board. And lastly, I want to thank my sisters for inspiration, Judy and Susie, who passed away at the age of 14 and 9 back in 1972. So I'm pleased in their memory to be able to do something to make sure that patients and families don't have to suffer from this disease like they did. So with that, I'm happy to have Danae open the lines and take any questions you may have.
All lines have been unmuted at this time. Does anyone have questions for Margie on her presentation?
I just want to say thank you for taking time to do this. It's been very informative, and we just hope we can keep moving forward with all the research that HCU Network America and HCU Network Australia has been doing.
Thank you. So I'm sure there were some technical terms I used that some of you may not have understood. Don't be afraid to ask. You know, I have 5 years of a pharmacy education that enabled me to learn most of them, but many of you don't have the luxury of that.
Hi, Margie. This is Ben Lewis. Just a quick question. You talked a bit about the cost of some of the treatments and compared them to other treatments that are out there in the market and sort of how payers treat those. Is there anything around sort of a trade-off between quality of life and the payments? I know it's a broad way to ask the question. I was thinking of it because we were covering the way that the healthcare payer system works with the fairly recent changes to insurance in one of my classes a couple weeks ago, and I'm wondering if there's an incentive for insurers to cover treatments like this because the quality of life is better.
Yeah, that's a very good point, Ben. Thanks for asking that. And it's something, I don't know if Tara's on the line. If she is, I'll ask her to also comment, that Tara has been a real proponent of measuring quality of life. And she has scoured the global literature landscape and talked to experts to see if anyone's really looked at the quality of life implications of having CBS deficient homocystinuria, and there really not any good studies, which is why our experts recommended that we try to get some done. There are some measurements in a natural history study, which I didn't mention because it's not a study of any therapy, but there's a natural history study that Orphan Technologies is sponsoring that has sites in the U.S. and in Europe because they want to understand how HCU evolves over time, which is a backdrop then for the information that regulators will be looking at when they approve these products. And so we do believe that over time, if we can demonstrate how the dietary restrictions and some of the symptoms of homocystinuria, how it decreases the quality of life, we do believe that's important information for payers, and especially as it relates to the dietary restrictions, because you could take a cynical payer, and I've met a few in my day, who would say, why should I cover this therapy at X thousands of dollars a year when patients can manage this by just controlling their diet? And that would be a very naive and dangerous view for a payer to have. And so we really do need to show them how these dietary restrictions not only impact quality of life, but how compliance is very poor in many patients with these dietary restrictions, meaning you never achieve that effect. So theoretically, it's there, but either it lessens your quality of life by the sacrifices you have to make, or you don't follow it and you don't get those benefits of the dietary approach. Is Tara on the line to add anything else?
Yeah,
She may not be.
Yeah, I'm Muffy. Hi, how are you? Well, I do know when the guidelines were being written up and obviously the literature was being looked at, Martina Zuma pretty much came up with the answer that there is no reported data on quality of life. I think it was something, obviously, that we've tried to work towards in terms of doing the survey that we presented in Prague initially, and then the most recent survey that we're starting to circulate out. So we hope to get some data in respect to that and publish it, but it's not something that's currently available.
Thank you.
Tara. Time for a few more questions, if there are any.
Hey, Margie, this is Mark Lewis. I have a quick question for you. As much as you can divulge, what's realizing Orphan is just on the cusp of dosing in their phase 1. Any idea generally on the timing of the duration of that study and when they may actually have some top-line data to share as a result?
Yeah, I wouldn't expect any data this year. I don't know the specifics of their protocol, but generally a phase 1 study, you wouldn't expect to see data for at least 12 months after you get, a cadre of patients into the study. I believe that their goal was to get, if it was 20 or 30 patients into the study. So I wouldn't expect to see anything, until at least the end of next year or maybe early the year after. I don't know if there's anyone on the line who would want to give a different answer, but probably not. just takes a while to enroll enough patients, to do all the measurements, and then to pull the data together. That's a typical timeline.
Great, thank you.
Any other questions? Great. Well, Danae, do you want to comment on what you're going to do with this recording?
Yes. So this webinar this evening has been recorded, and so will the questions be included. And we will have this on our YouTube channel by tomorrow morning, so you can look for it there, and we will also be sharing it on social media and via email. So once we get that uploaded and online, please feel free to share it with others in the community or with your family as well.
Okay. Thanks, Danae.
All right. Thank you, everyone, for attending tonight's webinar.
Danae.
Thanks. Thank you, Danae.
Thank you.
Danae.